So today we discuss reflex action and reflex arc. Reflex action and reflex arc. Now, before we go to discuss reflex action, first of all, let us understand what is a reflex or what are reflexes. So, what is a reflex? What is a reflex? Now, a reflex is an automatic response to a stimulus. So, what is a reflex? It is an automatic response to a stimulus. Now, first of all, what is a stimulus or what are stimuli? A stimulus is any change in the environment that elicits, that evokes a response from the body. Any change in the environment, whether external or internal, that evokes a response on the part of the body is called a reflex. Now, what is a stimulus? Stimulus is any change in the environment, as I already said, outside the body, in the environment, or inside the body, in the internal environment. And so long as the body is alive, so long as the individual is alive, it must continue to respond to these environmental changes or environmental stimuli. Now, reflexes can be of several types. First of all, simple reflex. Types of reflexes. Types of reflexes. One is the simple reflex. Now, what is a simple reflex? A simple reflex is one that engages only 2 or 3 neurons. And spinal reflexes that have their origin from the spinal cord. You must remember that the central nervous system is divided into the brain and the spinal cord. So, those automatic responses which have their origin from the spinal cord. Spinal cord is the posterior continuation of the brain. Brain is located safely in the brain box or skull. It is continued posteriorly as the spinal cord through the neural canals of the vertebral column. And any reflex that has its origin from the spinal cord is called a spinal reflex. And it is normally a simple reflex because it involves only 2 or 3 neurons, and these neurons are located in the spinal cord. So, and the other kind of simple reflex or spinal reflex. Spinal reflexes are usually simple reflexes. The second is the cerebral reflexes. Cerebral reflex. Now, what is a cerebral reflex? a reflex, of course, all reflexes are automatic responses. The body has no choice but to act in a particular way. So, they are automatic responses, and any automatic response that has its origin from the brain, from the head, brain, or from the cranium is called a cranial reflex or a cerebral reflex. Now, an example of a very simple example of a spinal reflex is that when our finger is pricked with a pin, immediately we tend to withdraw the hand. When the finger, one of the fingers is pricked with a pin, immediately we tend to withdraw the hand. Similarly, when we touch a very hot object, immediately we show a tendency to withdraw the hand. Now, these are two examples of simple spinal reflexes. Now, in the first diagram, as you can see, now the hand has touched a hot object. Now, when the hand has the immediate response is to withdraw the hand, but how this is done? Now, this change in the environment, that is the touching the hot object, is conveyed by this has to be an afferent neuron, because an afferent neuron is one that conducts impulses towards the sensory neuron. So, this is a sensory fiber, or it is a dendritic fiber. A nerve cell has two types of fibers, as we are aware. One is the dendrites. Dendrites usually they are afferent, AFF, afferent means sensory, and they conduct impulses towards the cell body. Now, the cell body is located in a dorsal, this is the sensory neuron located in the dorsal root ganglion. Now, a spinal nerve, this is the course of action, this is the reflex arc shown in a spinal nerve. Now, the spinal nerve has two roots, a dorsal root that is called the sensory root, and a ventral root that is called the motor root. So, as soon as we touch a hot object, or as soon as somebody pricks our finger with a pin, this sensation is carried by an afferent or dendritic fiber towards the cell body located in the sensory root. So, this is that sensory neuron, this is the dendritic fiber. Dendrites conduct impulses towards the cell body, and then this impulse is carried by an axonic fiber. Now, this is a dendritic fiber, this is an axonic fiber. This is axonic fiber is represented by A. So, this dendritic fiber carries the sensory impulse towards the cell body located in the sensory neuron, in the dorsal root or the sensory root, and then from the cell body it is transmitted, it is conveyed by the axonic fiber towards the, you see, if you take a section of the spinal cord, the matter is divided into an inner gray matter. The gray matter is butterfly shaped, where most of the cell bodies of the neurons are contained. That is why it appears gray in color. Now, this gray matter is produced into two dorsal horns and two ventral horns. So, this axon which has carried the sensory impulse from the hot object via the sensory neuron. It is carried by the axonic fiber into the gray matter. Now, in the gray matter, there is another neuron that is called the relay neuron or interneuron. Now, this relay neuron has again, so it forms synapses. It's dendritic fiber. This is the dendritic fiber. Dendritic fiber forms a synaptic junction with the axonic fiber of the sensory neuron, and so here that synapse is formed, and then it carries to, so its axonic fiber then carries this impulse to another motor neuron located here. So, this is that motor neuron. So, motor neuron, and its axonic fiber will supply the same muscle, effector muscle, that will whose contraction will result in withdrawing the hand. So, so this reflex arc involves only 3 neurons, one sensory neuron located in the dorsal root or sensory root, one interneuron located in the gray matter, and another motor neuron. It may be located here, or it may be located here also. It may be located here also. So, the interneuron connects the sensory neuron with the motor neuron, and the motor neuron similarly has a dendritic fiber and an axonic fiber. A dendritic fiber forms a synapse with the axon of the interneuron, and its axonic fiber of the motor neuron will carry the impulse to the muscle. This muscle is located in the same area of the hand where this impulse first originated. Contraction of these muscles will enable us to withdraw our hand. So, this reflex arc, simple reflex, spinal reflex, involves only 3 neurons, one sensory neuron, one interneuron, and one motor neuron. Several examples can be cited. Say, whenever we have seen, apart from these, whenever we have seen that whenever a food particle enters into, not into the esophagus, which is the opening of the alimentary canal, but into via the glottis into the respiratory tract, then immediate reflex is to cough it out. We undergo violent coughing reflexes to throw this food particle that has by mistake entered into the respiratory tract without entering into the alimentary canal. So, this is again a spinal reflex, coughing reflex. Again, reflexes can be categorized into, and what about the cerebral reflex? This is an example of a spinal reflex. Now, cerebral reflex, an example can be when strong light falls on our eyes show a tendency to close. Closing of the eyes when strong light falls upon them is an example of a cerebral reflex. So, cerebral reflex. So, cerebral reflexes originate from the cranium or the brain box or the brain or the so structures that originate that are present in the head elicit these cerebral reflexes. So, simple or spinal reflex and the other is the cerebral reflex or cranial reflex. An example is closure of the eyes, when strong light falls on them. Another distinction of reflexes can be unconditioned and conditioned reflexes. Unconditioned, another basis of division or another type of division is unconditioned and conditioned reflexes. Now, all these examples which we discussed just now, they are examples of unconditioned reflexes. And what are, so these are all examples of unconditioned reflexes which are till now discussed. Now, what is then a conditioned reflex? A conditioned reflex is because of it's a habit forming reflex. Habit forming reflex. Now, an example can be, say, when you are cycling. You are cycling maybe in a park, and your thoughts are somewhere else. That means, without thinking, because of the habit, you go on cycling. So, these habitual reflexes, habit forming reflexes are called conditioned reflexes. Now, these reflexes are developed by training. By training, we have developed this habit of cycling without being even aware of it. Mind is elsewhere, but the legs are moving, hands are also working, and so such habit forming reflexes that result from training are called conditioned reflex. Now, this conditioned reflex, the phenomenon was first developed by the Russian physiologist Ivan Pavlov, for which he was awarded a Nobel Prize. Now, what he did, while feeding a dog, he made an experiment on a dog. Now, when he used to feed the dog, he continuously used to ring a bell. Now, when the dog was not even provided with food, only some other time, when the dog was not provided with food, only the bell was rung, still there was salivation from the tongue of the dog. Because when food was provided, saliva was secreted from its tongue. So, associated with this provision of food was ringing of the bell. But when no food was supplied, only bell was rung at some other time, then the same salivation was observed, even if no food was provided to the dog. So, this is because the dog was accustomed to this ringing of the bell, it accustomed its salivation with the ringing of the bell. So, when there was no food, only bell was rung, the salivation took place. So, such reflexes which are developed by training, which are habit forming reflexes, are called conditioned reflexes. For the first time, it was invented by this Russian physiologist Ivan Pavlov. Next we come to, after reflex action, reflex arc, types of reflexes, conditioned and unconditioned reflexes, we next come to the conduction of nerve impulses. Conduction of generation and conduction of nerve impulses. Generation is an important topic. Generation and conduction of nerve impulses. Before we go to the generation and conduction of a nerve impulse, let us understand that this generation of a nerve impulse occurs in a receptor. Receptor. First of all, what is a receptor? A receptor is a modified nerve ending, modified enlarged nerve ending, which has the ability to generate a nerve impulse. Now, thereby all receptors, there are several types of receptors in the body, all receptors act as transducers. Transducers. What is a transducer? So, what is a transducer? So, a receptor is a transducer. Now, what is a transducer? A transducer is a structure which has the ability to change any type of stimulus, any type of energy, any type of stimulus into electrical energy. Any type of energy like say light or pressure or heat, any type of stimulus or energy is converted to electrical energy. So, a transducer therefore has this ability to change all types of energies into finally into one electrical energy, and then to carry this in the form of a nerve impulse. Now, what is a nerve impulse therefore? Nerve impulse is information that is conducted by the neurons from the transducer to say from one neuron to another neuron. Now, the nerve fibers usually carry impulses in the form of an electrical impulse. Now, electrical impulse, whatever may be the nature, it is converted to an electrical energy or electrical impulse. Now, then this electrical impulse is transmitted along the nerve fiber, and as we have already seen, dendrites usually carry impulses from the dendrites usually carry impulses towards the cell body, and axon carries impulses away from it. So, the nerve fiber that originated from the receptor, from the transducer, is a dendritic fiber. Now, this dendritic fiber then carries this electrical impulse towards the cell body. And from this cell body, it is carried towards the next neuron via the synapse by the axon. So, a dendritic fiber carries it towards the cell body, and an axonic fiber carries impulses I mean information practically it is information only that the neurons conduct, information in the form of an electrical impulse from the transducer via the dendritic fiber to the cell body, and from the cell body via the axonic fiber to the synapse. Now, we have seen that synapse is the junction place where information is exchanged from one neuron to another. So, from the synapse again after this exchange, again the information will be carried by a dendritic fiber, because only dendritic fiber can carry it towards the cell body. So, dendritic fiber to the cell body, and from the cell body again via the axon to the next neuron, and that is how all the neurons are joined with one another. Are they, of course, they do not come in any physical contact, but functionally all the neurons come together to form a neuronal network in the brain and in other places of the body. Via this neuronal network, information from the receptor is conveyed to the rest of the body. So, these receptors, now in this, let us devote 1 minute more. Now, this synapse across which the information was exchanged between one neuron to another. Now, this neuron can be called the presynaptic neuron, presynaptic neuron, and the other one which received it can be called the postsynaptic neuron, postsynaptic neuron. So, across the synapse, information in the form of an electrical impulse originated first in the receptor is transmitted from a presynaptic neuron to a postsynaptic neuron, and in this manner it can be conveyed to a host of other neurons in the neuronal network. So, next we come to discuss one or two concepts which are important in the understanding of the conduction of nerve impulses. First of all, what is a membrane potential? What is a membrane potential? Also called the resting potential. Resting potential. Now, all cells in the body, say this is the fiber, nerve fiber. It may be dendritic or axonic, does not matter. So, this is the nerve fiber. Now, in a nerve fiber, the nerve fiber is or has a membrane potential. That means the outside of the fiber is said positive. So, this is the outside of the fiber, nerve fiber. It may be a dendrite or it may be an axon. So, the outside is said positive, and the interior of the nerve fiber is said negative. Outside is positive, inside is negative. Now, this state of the nerve fiber, the outside is positive, the inside is negative, is because of the unequal distribution of the ions, unequal distribution of ions. Usually we will only discuss those ions which are most important from this point of view. So, unequal distribution of ions, mostly sodium ions and potassium ions. Now, it is seen that potassium ions are present in much greater concentration inside. Potassium ions are present in greater concentration in the interior of the fiber, and in the outside there is a greater concentration of sodium ions. So, sodium ions are present in greater concentration outside the fiber. Now, when there is a concentration difference, these ions tend to diffuse across their concentration gradient. Sodium ions tend to diffuse into the interior of the fiber because of their greater concentration outside, and potassium ions show a tendency to diffuse out of the nerve fiber because of their greater concentration inside. So, this diffusion, this is called a diffusion from a region of higher concentration, flow of substances from a region of higher concentration to a region of lower concentration. Now, in case of potassium ions, the higher concentration is present inside the fiber, and in case of the sodium ions, it is present in greater concentration outside the fiber. But and this is the cell, it is the nerve fiber membrane. But what happens? Nerve fiber membrane. But this diffusion of ions occurs through ion channels. Ion channels are present in the cell membrane or the nerve fiber membrane. Now, ion channels for potassium in the membrane are more than sodium ions. And again, hydrated, because these ions are not dry, they are hydrated. Hydrated sodium ions have a larger size than hydrated potassium ions. So, sodium ions cannot enter into the interior of the fiber for two reasons. One is their hydrated size is more, secondly is larger, secondly there are less number of sodium channels across the membrane. Potassium ions tend to show, they have a lesser hydrated size and more concentration inside, and they have many channels in the cell, in the nerve fiber membrane. So, potassium ions show this tendency of continuously leaving the interior of the fiber to the exterior, as a result of which the interior becomes a shade negative. This minus sign indicates in the interior, shows that the interior of the nerve fiber, interior is negative, and the exterior is positive. Why? Because first of all, number of sodium channels in the membrane are less. Secondly, hydrated sodium ions are larger in size, so they cannot enter freely into the, although they show this tendency to diffuse, but because of the non-availability of channels in the cell membrane and because of their larger hydrated size, they cannot enter into the fiber. So, they are held in a greater concentration outside than the potassium ions, which are present as a result in a greater concentration inside the fiber. And these potassium ions, because of their larger number and availability of these channels across the cell membrane for potassium in larger number, potassium ions tend to always leave the interior. Potassium ions always tend to show this tendency of leaving the interior. Now, this tendency of the potassium ions to leave the interior makes the interior negative. You see, the sodium ions cannot enter into the fiber. Potassium ions always show a tendency to leave the interior. So, this tendency, because of this tendency and because of the presence of more channels across the membrane, potassium ions continuously show this tendency of leaving the interior, as a result of which, so because of these two regions, the interior becomes a shade negative, then the exterior. Exterior is a shade positive, because sodium channels are less and sodium ions are larger in size. Now, this potential difference that exists outside is positive, inside is negative, has been measured by a pair of electrodes, one of which was kept on the surface, another was inserted into the fiber. When the two electrodes were connected to an oscilloscope, immediately it showed a minus sign. So, this showed that and this difference in the potential has been measured at minus 70. Potential difference is measured in volts or millivolts, and this difference between the inside and the outside is minus 70 millivolts in case of the nerve cells. In case of the nerve cells have a potential difference or a membrane potential of minus 70 millivolts. Now, similarly, other cells also show different membrane potentials. Muscles show skeletal muscle minus 45, visceral muscles is minus 50. Now, this is the membrane potential or resting potential. I will come to that, why it is called resting potential. Erythrocytes, similarly, erythrocytes, you know what erythrocytes are. RBCs are also known as erythrocytes. Erythrocytes have a membrane potential of minus 12 millivolts. All these are in millivolts. And astrocytes, one of the glial cells in the brain, if you remember, apart from neurons, there are astrocytes. In fact, the number of astrocytes is much more than that of the neurons. And astrocytes have a membrane potential of minus 80 to minus 90 millivolts. Now, this is what is known as, this is the membrane potential. This is also called resting potential, particularly in excitable cells. Excitable cells, what are excitable cells? Excitable cells are neurons and muscle cells. Neurons and muscle cells can exist in two states, a resting state as well as an excitable state. So, resting state, the membrane potential is minus 70 millivolts, and the neurons can change into an excited state. Or there are different terms for them. This is also called the polarized state, because the outside is a shade positive, the inside is a shade negative, outside positive, inside negative. So, this is called a polarized state. And in case of the excitable cells, they have another state also, that is called the depolarized state. And because these cells can exist in two states, a resting state or a polarized state, and an excitable state or a depolarized state. So, these abilities, these characteristics are found particularly in neurons and in muscle cells. All other cells have more or less a normal resting potential that does not change from a resting to an excited state. But since these are excitable cells, they show a tendency to pass from the resting state to an excited state, or from a polarized state to a depolarized state. Now, what happens during the passage of the impulse, during the passage of the information from the transducer, from the receptor towards the cell body? Now, when the information is conducted from the sensory receptor towards the cell body by a dendritic fiber, momentarily, briefly, there is, so this is the information that is to be conducted, direction of information conduction, and during the state of information conduction, suddenly this polarized state changes into depolarized state. That means minus 70, which it was, membrane potential was minus 70 millivolts in the resting state. During conduction of the impulse, it changes into the, so this is the polarized state, polarized to depolarized. So, during the conduction of the information in the form of an electrical impulse, suddenly the sodium gates, the floodgates for sodium are opened, as a result of which sodium ions now can enter, the interior of the fiber. The channels were closed earlier in the polarized state, but in the depolarized state, which is characteristic of the information conduction, suddenly the gates for sodium are opened, and sodium ions can now enter into the interior of the fiber in larger number, as a result of which the polarized state of minus 70 millivolts now changes even to plus 40. That means, so much of sodium ions enter into the interior of the fiber that the interior now becomes not only depolarized, it becomes even, goes to even the plus side. So, minus 70, it changes to plus 40, minus 70 to plus 40, because of the sudden entry of sodium ions. Now, this is, so in this state, it is called, so earlier when it was not conducting an impulse, it was called a resting potential. Now it is called an action potential. Action potential. So this action potential results from the sudden entry of the sodium ions into the nerve interior of the nerve fiber. Now it is called the action potential. Now this state of action potential then spreads along the fiber, towards the cell body. As the area in front changes from the polarized to the depolarized, the area left behind is again repolarized. That means again it changes back from the depolarized to the polarized state. How this happens? So, as the area in front, how the information spreads, how the information is conducted. Information is conducted in the form of a potential difference across the fiber, and that is because of the sudden entry of the sodium ions. The fiber becomes now from -70 it changes to +40, and this depolarization spreads along the fiber. So the information spreads means the information spreads in the form of a depolarization. Now this depolarized state spreads, and as it spreads, the area in front of it is depolarized. Area left behind must be repolarized, must go back to again -70, otherwise another impulse cannot be conducted. Now this depolarization to repolarization, depolarization to repolarization, that means +40 to again -70 occurs by the working of a sodium potassium pump. Sodium potassium pump. Now what is that sodium potassium pump? Now in the cell membrane of all cells, sodium potassium pump, in all cells in their membrane there is a sodium potassium pump in operation. In fact, whatever food, most of the energy that we derive from the foods that we eat is utilized in maintaining this sodium potassium pump across the membrane. And how does this sodium potassium pump operate, and how it brings about again a change back to the polarized state? Now this, say, this is that sodium potassium pump. Now this operation of the sodium potassium pump needs energy in the form of ATP. So when ATP is present in the cell, so this is the interior, and in the interior ATP is present. ATP, the energy currency, when the cell wants to perform any function, just as when we go to the market to purchase something, we spend a few coins or few notes for the substance that we purchase from outside. Similarly, the cell has a currency of a different kind. It has this ATP as its currency which it utilizes. So when this ATP is hydrolyzed to ADP and PI, energy is released. Now this energy is utilized. The moment the ATP joins with this pump, the pump has two sides, an outside and an inside. So from the inside, when an ATP molecule joins its binding site from the inside, then the energy is provided, and in this energized state, the pump has two binding sites for potassium, in this energized state, it has 3 sodium ions, sites for 3 sodium ions accessible from inside. So, now sodium ions have entered in large number, the sodium ions must be again driven out from the interior, so that the state can change from the depolarized to the repolarized state. So, this sodium ions are expelled to the outside by the sodium potassium pump. How? Now, when the ATP binds itself to the sodium potassium pump from inside, the binding sites for 3 sodium ions now become accessible. So, 3 sodium ions now can attach, 3 sodium ions can now attach to their binding sites accessible from the inside. Now, this binding of sodium ions from the inside of the fiber changes the conformation of the pump, changes the conformation. Conformation means shape. Shape of the pump, sodium pump, is changed, and in this changed state, the sodium ions are left outside, and from outside, potassium ions, 2 potassium ions are again 2 potassium ions are again picked up in this changed state of the pump. So, in exchange for 3 sodium ions, 2 potassium ions are picked up from outside, as a result of which sodium ions now are expelled out. Now, you will ask a question here. Now, when the nerve impulse was not conducted by the nerve fiber, what was the role of this sodium potassium pump? Now, this sodium potassium pump again has the same function, that of expelling the sodium ions and in exchange with potassium, so that the interior always was kept low in sodium. So, this diffusion and the availability of channels, as well as the operation of the sodium pump, were responsible for keeping the interior always low in sodium and high in potassium. Now, mammalian axons have an interesting ability. Mammalian axons, so let us say that this is the mammalian axon. So, this is the direction in which the electrical impulse spreads in the form of a depolarization. As the area in front becomes depolarized, area left behind is again repolarized. Now, the axonic fiber, if you remember, has a coating of myelin. So, this is the axonic fiber, and this is the coat of myelin. Coat means covering of myelin. Now, this myelin, if you remember, is synthesized by the oligodendroglia, kind of glial cells present inside the brain, oligodendroglia, and outside the CNS by the Schwann cells. So, whether oligodendroglia or Schwann cells, these are responsible for secreting this myelin sheath. Now, myelin sheath acts as, just as in a wire, electrical wire, you have an outer coating that prevents leakage of current. The current flows through the the electrical fiber, but outside you have a non-conducting covering in case of the electrical wire. Similarly, also here the outside there is this myelin sheath. This myelin sheath helps to prevent leakage of the leakage of current or the electrical impulse, as in this case, the information that spreads along the fiber in the form of an electrical impulse. So, this is that myelin sheath. It prevents the leakage of current, but this myelin sheath is not continuous. As you can see in this diagram, the myelin sheath is interrupted at places that are called the nodes of Ranvier. Nodes of Ranvier. Ranvier is the name of the scientist who for the first time. So, this myelin sheath is interrupted at places by these nodes of Ranvier. So, because of this myelin sheath and the nodes of Ranvier, the depolarization, the electrical impulse need not traverse the whole of the fiber. It simply can jump from one node to another. It can only, the nerve impulse can jump from one node to another. Now, this type of conduction of the nerve impulse in the mammalian axons is known as saltatory conduction. Saltatory conduction. Saltatory means it's a kind of jumping mode of conduction of the nerve impulse. Instead of passing along the whole length of the fiber, it simply jumps from one node to another. As a result of this myelin sheath and saltatory conduction, mammalian axons are able to conduct impulses much faster. So, faster, what is the advantage? Faster rate of conduction. Faster rate of conduction of the nerve impulse, and up to even 100 meters per second, even up to 100 meters per second. Nerve fibers in mammalian neurons can conduct impulses because of this myelin sheath and nodes of Ranvier, which are lacking in the other lower vertebrates and invertebrates. Next, we come to the synaptic transmission, generation and conduction. After generation and conduction of the nerve impulse, we come to now what happens at the synapse or synaptic transmission. Synaptic transmission. Now, we know that an axon ending is enlarged to form a synaptic knob. This is that synaptic knob. This is the axonic fiber, axon ending. So, the axon ending is enlarged to form a synaptic knob. So, the impulse has arrived at the axon ending. This is that synapse. The space between, there is no actual union, there is a small gap of about 200 angstroms. This is what is known as the synaptic cleft. Synaptic cleft. That means there is a small gap which is about 200 angstroms, synaptic cleft. Now, the moment the nerve impulse arrives at the axon ending in the synaptic knob, you see, as you can see in this diagram, number of synaptic vesicles are there, small pockets of a chemical substance. Small pockets, synaptic vesicles are nothing but small synaptic vesicles are nothing but small pockets of a chemical substance that is called the neurotransmitter. Neurotransmitter, a chemical substance that is called a neurotransmitter, which helps to transmit impulses from one neuron. This is a part of the presynaptic neuron. This has to be a dendrite. So, impulses are transmitted from the axon to the dendrite, exodendritic. This is an exodendritic transmission. So, the neurotransmitter or nerve transmitter is a chemical that is contained in the small pockets that are present at the present in the synaptic knob. Also can be seen in the synaptic knob a number of mitochondria, because mitochondria are required for providing energy for this transmission of nerve impulse. So, then there is the synaptic cleft, and then opposing this synaptic cleft you have the dendrite. So, this has to be a dendrite which will then carry impulse towards the cell body. Now, this is the dendritic membrane. So, between the axon ending membrane and the dendritic membrane, there is a small gap, synaptic cleft. Now, in this cleft are present calcium ions. Calcium ions are necessary. These calcium ions, so here calcium ions are there. Calcium ions are picked up into the synaptic knob, because calcium ions are required for the release of the neurotransmitter from the vesicles. So, neurotransmitter is contained in the vesicles. The vesicles then come near the membrane, then join with them, and then the area of joining, as you can see in this diagram, bursts open, releasing the neurotransmitter into the synaptic cleft. Now, this is made possible by the calcium ions which are there in the synaptic cleft, which are picked up by the synaptic knob. So, now this neurotransmitter in most mammalian neurons is acetylcholine. Now, this acetylcholine is a neurotransmitter which is present in the synaptic vesicle, and this acetylcholine is released into the cleft when the nerve impulse reaches the axon ending, and there is the entry of calcium ions, and calcium ions are required for a process called exocytosis. Calcium ions bring about this exocytosis, and because of this exocytosis, during this exocytosis, the vesicle membrane and the axon ending membrane fuse, area of fusion breaks open, releasing the acetylcholine into the synaptic cleft. Now, then this this acetylcholine crosses this cleft. So, it is a chemical, now this acetylcholine then crosses the cleft and binds itself to specific receptors. These are the receptors depressions as you see them on the dendritic membrane. They are the receptors for this acetylcholine, where acetylcholine binds itself. Now, binding of acetylcholine to its receptor sites in the dendritic membrane initiates again that depolarization. So, the dendritic membrane therefore becomes depolarized, and this depolarized state then spreads along the dendritic fiber towards the cell body of the next neuron, that is, as we have named it as postsynaptic neuron. This is a part of the presynaptic neuron. Now, this dendrite is a part of the postsynaptic neuron. So, impulses are conducted therefore from the presynaptic neuron to the postsynaptic neuron across the synaptic cleft by this acetylcholine, so which is a chemical transmitter. So, passage of nerve impulse across the synaptic cleft is therefore not electrical, but chemical. Then this acetylcholine binds and to the receptors, binding again initiates the depolarization wave from the dendritic membrane. Now, such neurons which release acetylcholine from their